Welcome to an Achieving Science video. This video is going to look at growing bacteria. First of all, some background. Bacteria can be grown in what's called a culture medium. This can either be a nutrient broth solution or solid agar jelly. In the picture, we've got solid agar jelly in a petri dish. The solid agar jelly contains carbohydrates, minerals, proteins and vitamins. Here you can see in the picture bacteria that has grown on these agar plates. On the left hand side you can see the small visible colonies, and on the right hand side you can see where they've been spread out to give a much more even coating. When growing microorganisms in school then, there are some key rules. The first one is that we cannot keep them above 25 degrees Celsius. This prevents any harmful bacteria from growing. Secondly, when sealing the plates, we mustn't seal them completely. We must always allow air to get in, and therefore preventing any anaerobic pathogens from growing. Thirdly, when labelling up the plate, we always label around the edge so it doesn't obscure our vision. And we must always label it with both the bacteria that is grown and the date that it was plated. And finally, when storing these plates, we must always store them upside down. That prevents any of the condensation that forms from falling onto the surface. Now, in order to culture some microorganisms in the lab, we will require a petri dish, usually a plastic dish with a base and a lid, and we'll require an inoculating loop, which is a thin piece of wire with a small loop, as you can see on the end. We want to prepare uncontaminated cultures. This means that we do not want any airborne pathogens or microorganisms to get into our plate or anything that's on the surface, so we need to be careful about what we do. So we can avoid this by first of all disinfecting the area, wiping down our area with disinfectant. We can work close to a Bunsen burner. We can also sterilise both the petri dish and the culture medium by heating it to a high temperature. We can place the inoculating loop in a hot flame where it will glow red, but we must always ensure that once we've heated it and killed off any microorganisms on it, that we do allow it to cool before using it, because we do not want to kill any bacteria that we want to transfer from a broth to an agar plate, for instance. When lifting the lid of the petri dish, we'll only do so when needed, and we'll only lift it slightly to avoid any airborne pathogens, microorganisms getting in. We'll also make sure that once we finish with it, it is sealed, but remember, not all the way around, only locations around the edge to make sure it is sealed, but also to allow air to still get into the dish. And also again, store it upside down to prevent any condensation from falling onto the surface. Now, in order to test some antibiotic discs then, let's see the effects that antibiotics have upon bacteria. To start with, we need to have a petri dish that is prepared. This means that the hot agar jelly has been poured into the dish, it's been allowed to cool and set. We then covered that jelly once it's cooled with the bacteria and made sure there is an even covering of bacteria. Once this is done, then we can begin to test some antibiotics. First thing you do is you take some paper discs and you place them into some antibiotics or disinfectants and leave them just so the antibiotic or the disinfectant can penetrate that paper disc. You'll then transfer those discs using tweezers or forceps onto the agar plate, spreading them out evenly so they don't overlap. We'll also need a control disc. Instead of placing the paper disc into a disinfectant or antibiotic, we'll actually place this into sterile water. Then we can see that the effect was caused by the antibiotic rather than the paper disc itself. Then, once that's all set up, we'll leave it for 48 hours at a temperature around 25 degrees Celsius, and then we'll see the results. Let's have a look at some results. Here you can see on the slide that we used several antibiotic discs, which are these again paper little discs, and you can see on here then this cream area is where bacteria still remains, and these clear zones, which are referred to as inhibition zones, are where the bacteria has been killed. So this antibiotic disc has killed a lot of the bacteria around it and it is the most effective, as where something like this one is ineffective. If it has killed anything, it's very, very minimal that it's killed. So the effectiveness is therefore seen by the clear zone. We can either do this by placing it on a piece of graph paper, or we can measure the diameter and work out the area that has been killed. Because these are circular, we would therefore use pi times r squared. Where we've got the largest the inhibition zone, the more effective that antibiotic or disinfectant is. Let's have a look at some measurements then. So here we've got a agar plate. You can see the bacteria in cream, and you can see the clear zone around the antibiotic disc. There are two methods to this. Either you can count the number of squares individually, so 1, 2, 3, 4, 5, 6, 7, 8, or you can use the diameter. So we've measured that as 8 squares. If we look at the key, and the key said that a square is 1 centimetre, we know that that would therefore be 8 centimetres. If we know the diameter, then we can use the equation pi r squared, so that half of the diameter would therefore be 4. 4 squared is 16. 16 times by pi would give us an area of 50 centimetres squared. This one, again, we could count every square. We have problems here where we haven't got a complete square, so we could use the procedure where anything that is bigger than a half a square, we count as 1. Anything smaller than half a square, we wouldn't count. So we could use that method. Or, again, we could count the number of squares across. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 squares across. That would be our diameter. Again, look at the key. If it's 1 centimetre for 1 square, we can use pi r squared, and we'd come out with 78.5 centimetres squared for this one. The final example then is a smaller one. Again, you've got to decipher whether this is a full square here, so are we saying this is 1, 2, 3.5, or is it a little bit less, a little bit more? There will be a little leeway with your answer. I went for 3.5 centimetres, so therefore came out with a 9.6 centimetre squared clear zone with this one. So this was the least effective of the 3 different antibiotic discs.